Hello, my name is Elliot Andrew, and today I'll be talking to you all about testing the relationship between primary productivity and animal abundance. So for my experiment, my team and I researched and looked at data for several animal species, such as this raccoon. And I don't know about you all, but I can certainly relate to the excitement this raccoon is showing when performing experiments and looking at animal data. For my research project, my group was asked the question, does primary productivity affect animal relative abundance? So what is primary productivity? This is the rate at which plants and other photosynthetic organisms produce organic compounds in an ecosystem. Relative abundance refers to the percent composition of an organism of a particular species relative to the total number of organisms in the area. We also know that the latitude diversity gradient, which is the global patterns of species diversity, are thought to reflect the energy available in an environment. This energy is typically described as the gross primary product, or GPP, which is a measurement of plant photosynthesis. We also know that the many individual hypothesis suggests that higher levels of GPP support more individual organisms. This then promotes the evolution of species diversity. On the other hand, the factors that drive the relationship between GPP and the latitude diversity gradient are not well understood or researched. The credibility of MIH hypothesis had gotten mixed empirical support and has only been tested for a few taxonomic groups. This is why we are going to test this hypothesis for a wider range of species. Understanding these factors are important to understand how species diversity works. By understanding the drivers for species diversity, we're able to understand evolutionary history better, make wiser decisions concerning environmental issues, and it aids in development planning. Our objective for our experiment was to test the many individualized hypothesis with mammals as a larger sample study group, as previous studies had only really looked at invertebrates and birds. We had two main hypotheses for our study, which were that the relationship between primary productivity and herbivores will have a strong positive correlation. This is because they have the most direct reliance and effect on vegetation. Our second hypothesis was that the relationship between omnivores and carnivores and primary productivity will be present, but will be at a weaker correlation. To carry out our experiment, we used animal occurrence data from the 2019 Snapshot USA Mammal Survey, which looks to document the abundance and occurrence of animals across the United States. This used 1,513 camera trap sites in 107 sub-projects that were located across the continental USA. We then compared the measure of cumulative gross primary product to the relative abundance of mammals, which we broke down into the groups of carnivores, omnivores, and herbivores. After this, we used the detection rate as a measure of relative abundance of all mammals by dividing the total number of detections for each dietary classification by the number of days the camera traps were active. All this would not be possible without the help of the zoology, fisheries and wildlife, conservation biology, and animal science departments of the North Carolina State University in Raleigh. So through this study, we found that there were no relationship between GPP and carnivore abundance due to our R-squared value being 0 and our p-value being 0.98. We also found that the omnivore abundance was slightly positive correlated with GPP with an R-square value of 0.02 and a p-value of 0.11. Interestingly enough, we found that the relationship between GPP and herbivores was actually slightly negative with an R-square value of 0 and a p-value of 0.78. Based on the lack of correlation between GPP and all the groups of mammals, we concluded that our data does not support any relationships and that areas with higher GPP do not necessarily have a direct influence on relative animal abundance and species diversity for the locations that we tested across the United States, as suggested by the many individuals hypothesis. However, it is still possible that there are still relationships between GPP and more specific species. In the future, we believe that studies investigating MIH should examine other factors that affect the total number of individuals in communities, such as nutritional content and the ability of a species to forage, species generation times, and population densities. So I hope you all learned a little bit more about how primary productivity and the relative abundance animal are intertwined, and I thank you all for listening.